Hello and welcome to Biofilm Podcast, a show that brings you to the forefront of biomedical research, biotech, pharma and healthcare fields, and the professionals behind it. I'm your host, Pavel Rozhov. Please subscribe to my channel and hit the notification bell to stay up to date. Let's get started. All proteins essentially are comprised of amino acids, and they are interlinked together in this essentially polyamino acid chain. Now, these long chains of amino acids, they don't just exist as beads on a string, if you will. They exist in this sort of very intricate shapes, and depending on the protein, the shape of that chain of amino acids would be different. This is one of the fundamental principles of protein structure, where the sequence of those individual amino acids that comprise the length of your protein actually defines this very unique and very complex structure of the final protein. Now, that structure of the protein is super critical for this protein to be able to do what it's supposed to do. The classical example is something like a hemoglobin, the protein in our blood that is transporting oxygen and allows our cells to actually obtain the oxygen that is vital for our growth and sustainability of our organism. The shape of that molecule is unique in such a way that it allows for the oxygen to be accepted and carried and transported over. So and when, for example, you have that protein misshapen, it no longer has that function. So people have diseases. And this is true for all the proteins. Their structure, this very unique three-dimensional shape that they have, defines how efficient they are in carrying out their tasks. And in what happens in diseases, by and large, these proteins may have some sort of alteration in their structure, which directly affects how they're functioning, and that's what causes a disease.
That's great. One thing you mentioned that maybe not all the students know about is amino acids. Could you explain a little bit more about amino acids and what that means and how they fit together?
Yeah. So amino acids are small. I mean, small is everything is small when we talk about proteins or cells, but amino acids are small molecules which are organic molecules, meaning they are found in living organisms. This is we have about 20 amino acids, 20 types of amino acids from which all the proteins are comprised of. So you think you have 20 different types of amino acids that you can string together on this think beads on a string type scenario in any order that you see fit. And basically that order and the diversity of those 20 different amino acids actually yields this specific shape of the protein because of the way that they look, because of the way they interact with one another on that string. And this essentially allows them to create this phenomenal diversity of shapes, sizes, and things like that. But another thing I want to sort of elaborate on is the amino acids don't just come out of nowhere. When we talk about amino acids and when we talk about proteins, we need to talk about another side of that coin, which is DNA, where all of our genetic information is stored. And this is how we carry the genetic information from person to person, from organism to organism. DNA actually codes for the sequence of those amino acids in the protein. And this is what allows one to replicate all those different proteins that we find in a banana or in a bacterium or anywhere else. We share a lot of those same proteins for the same tasks. Yeah, so that's just kind of my short answer.
And my company, we work a lot with peptides. Do you think you could explain, because sometimes people have heard about peptides in relation to maybe a skin cream, but that isn't what we are talking about in science. So could you explain peptides and protein and amino acid and how they all...
Yeah, so the quick answer is the amino acids can be strung up in a string, and depending on the length of the string, you can sort of differentiate roughly between peptides, which are short chains of your amino acids, and full-fledged proteins that are something like a few hundred or a few thousand amino acids long. Peptides, you would think of peptides as something between like 10 to 15 to 50 maybe amino acids long. This would be like the length of those peptides that we were dealing with. And they're different in terms of peptides would have simpler shape to them, and proteins are much more complex in terms of how that sort of string is sort of crumped together in this sort of knot, if you will. Yeah, peptides are a little bit easier to deal with.
Yeah, definitely. Would you explain a little bit more about how they are manufactured and made? Because maybe some of the kids have heard about manufactured DNA and synthesis.
Yeah, so while I'm not an expert on exactly how peptides are manufactured per se, especially without going into too much detail, there's different ways you can go about manufacturing DNA or proteins. So let's start with DNA. DNA can be manufactured. First of all, it's manufactured by all of us at this instant, right? We are living beings. Every cell in our body can manufacture and replicate DNA. We leverage this property of living cells to synthesize new DNA very heavily across the life science spectrum. We rely on bacteria, for example. In my laboratory, we rely on bacteria to synthesize new DNA for us. At the same time, when we talk about, say, proteins, we rely on the same bacteria to not only synthesize the DNA that we want, but that DNA would usually code for the sequence of those amino acids that we make the protein out of. So we rely again on the bacteria to actually generate and manufacture those proteins for us that we then use to study them. So we heavily rely on these organisms as those mini biological factories, if you will, for production of DNA or the proteins. If we talk about industrial scale in sort of larger quantities, there's other methods to do that do not necessarily involve using organisms for that purpose.